Hello, and welcome to this webinar titled Evolve Manufacturing Processes by Implementing Disruptive Technologies, hosted by Biopharma Asia Magazine and presented by Yuyi Shen, Associate Director and Head of Development and Manufacturing at Bolt Bio, and John Cyganowski, Director of Manufacturing Sciences and Technologies Downstream Americas for MilliporeSigma. My name is Stephen Edwards, and I will be your moderator. Now, please allow me to introduce our first presenter. Yuyi Shen is the Associate Director and Head of Process Development and Manufacturing at Bolt Biotherapeutics. She also served different scientific roles in process development and manufacturing science departments at various biotech companies, including Zoma, Bayer, BioMarin, and Grifols. She holds a PhD degree in chemical engineering from the University of California in Davis, California. Yuyi provides scientific leadership with a specialty in process improvement, scale-up, technical transfer to bioprocess manufacturing in cGMP environments. She is a strong subject matter expert in designing robust processes for protein purification with monoclonal antibodies, antigen and viral-like particles, etc., ensuring critical quality attributes are achieved. She had solid troubleshooting and problem-solving skills and encouraging innovation technology to achieve high efficiency and better quality. I'll now be handing over to our first presenter. Welcome, Yuyi.
Hello, everyone. Thank you, Stephen, for the nice, kind introduction. I hope everyone has a good week. So I'm very happy to present today's topic about the involved manufacturing process by implementing disruptive technologies. Today's agenda including a brief introduction, and we're going to talk about the challenges and the major drives for the process development by the innovation technology. Then I will present a few case studies, including the process efficiency and how to reduce the cost using the innovation technology, and then I will finish up with the QBD strategy to minimize the risk, where we balance the needs for the quality, cost, and innovation. So when process scientists try to implement the innovative technology in the process, and then when they talk to people about making the change, this is pretty familiar questions that you get from the people that they want to implement those changes. At the CMC area, they will say, we'll deal with the improvement when we have time. Some people will be saying, can we lock the process right now and think about this later? And some people will say, we don't have a budget now, and we don't want to take any risk. So all this question is very legitimate, and they come from a true reason, because biotech is a heavily regulated environment, and it is controlled, and the culture is conservative. We want to keep patient safety as our first priority. So there's a risk concern about any changes to the process that can invoke any product quality and the process robustness to ensure the quality is not at risk. Also, biotech company is a very highly expensive industry compared with other industries. So we always have a problem, running into a problem with scale resource. We are limited by the time. We want to speed to the market. We have very few people, very few scientists always, and we also have very limited funds that we want to use wisely. So that is when we implemented try to implement the innovation technology in the process, and all this roadblock we will be seeing. So some people will be asking, what is truly considered as a disruptive technology? My view of 2 years is the disruptive degree is improving and increasing based on the stage of the product life cycle. At early R&D stage, we have lots of new ideas, very brilliant innovation technology we want to implement. But at that time, it's not considered as disruptive because it's not quite implemented to the final process just yet. And with the process development, we were able to screen out some very innovative technology and very practical and try to implement it so the disruptive degree will be increasing. And by the end, we reached to a commercial manufacturing. There's only very few innovation technology got implemented, and that degree of disruptive is very high. So the roadblock we often see, including the concerns from quality, the risk, from regulatory, from compliance and control. Also, it depends on the cultural shift between different functional group, between R&D group to PD group to manufacturing. The culture is getting more and more conservative in a more and more controlled environment. We also, like I said, we have very limited budget, try to implement something new. But the major driver for the innovation technology is still there. It is also coming from the quality concern, because we always try to improve our product quality, try to improve the purity, and we try to make our process more automated. That requires new technology. We try to reduce the cost. By implementing the technology in the end, we often see that the cost reduced. And then we also try to make the process more robust and more scalable with high yield, high efficiency. We can achieve the timeline better by implementing a high efficiency process. And the company try to remain competitive even after they launch a commercial product in the market. Our regulatory FDA in US also are very encouraging the innovative these years. So they will design some breakthrough therapy and a fast track designation, which requires a very expedited pathway. And to reach that pathway without implementing any innovation, that's kind of being very challenging. So that also requires us to pay attention to all the innovative technology these days. So first, I want to highlight a few of the areas that currently we have that including lots of innovation technology already. So basically, for the process development, we are thinking upstream and downstream. For the upstream, when we use a cell culture, there's a perfusion bioreactor considered as a pretty novel technology since the past 10 years, and it requires also very innovative cell retention devices, so the ATF cell retention versus the gravity cell retention device. Also, we are improving technology on the microfiltration side so that we can take care of the high titers and the high cell densities. For the bioreactor, instead of using the stainless steel, we implement more and more with the single-use technology. And the biotech with the process improvement on the feed and the medium, we were able to achieve pretty high titer. And also some things the bioreactor small scale process improvement, it requires a long time to do the cell culture and to do the screening. So sometimes the modeling also comes in place to replace all those tedious work on the bench scale experiment. For the inclusive cell, the lysis and the technology to disrupt the cell is also evolving. So we start with dynamill, microfluidizer. Recently, we also see some ultrasonic small scales for the lysis step. Also, again, for the cell clarification, we use the microfiltration. We use more discus continuous centrifugation. And also we combine it together with the depth filtration technology. For the downstream this year, we saw the continuous chromatography. And people are intended to use some single-use membrane to work with the resins. Very traditional way we try to purify our protein. And some of the non-chromatography separation techniques is also come back and also help with the further purification on the downstream. For the continuous chromatography step, also we see more and more PAT technology is used to analyze the in-process sample so that we have more robust control. Also, there's lots of emerging areas from the new therapy that opens more opportunities for the innovation. For example, we are seeing cell and gene therapy, more and more new companies formed on that area, so requires very brand new, not brand new, but more innovative technology in the process and manufacturing, because they're different scale. Also, we saw more next generation therapy that requires a write over of the new process. We see more biosimilar and biobetter to help this product remain competitive in the market. They also asking for innovation technology to improve the process efficiency and reduce the cost. Some of the example we've been seeing is for the cell and gene therapy. We saw manufacturing scale model instead of scaling up. We are seeing more and more small modular facility design that to accommodate the scaling out modules. It needs a flexible design. Also, the process requires lots of automation because right now, instead of we are doing a large scale, we basically want to have a mini scale with optimal close to control the process. So that's more, and the more technology we will be seeing from different scientists and different companies, that's to accommodate the needs for those new areas. Today I'm going to talk about some real case studies from my past life, from my past career, and that includes two case studies. One is the viral-like particle purification process, which implemented the high throughput single-use automation. And I also talk about a little bit of the cost reduction by screening the MAD chromatography step. So for the viral-like innovation technology, we actually implement about 6 innovative technologies. This is a good table that summarizes all the tools, technology tools that we use, what's their application, and what enable us to do for the process improvement. For example, the depth filtration, it's the first time we use a depth filter for the yeast cell purification because that's a very challenging cell line, and it's inclusive cell line, so there's a lot of cell debris after lysis. So we were able to work with vendor that enables the continuous purification. For the chromatography DOE steps, so we were able to use the buffer pro and the 2D chromatography that minimize our sample preparation and operation difference and minimize all the buffer preparation. As some of the scientists is doing DOE all the time, you will realize how much prepare work to making buffer with different pHs and samples with different conditions. And with this automation technology, we only need to prepare, we don't need to prepare samples, the machine will prepare samples and buffer for us. And we also implement at a large scale, pilot scale when we generated materials, so we implemented the inlet filter and the single-use membrane technology aligned with the clarification depth filter. So it comes with one train, and it basically significantly reduces the process time, and we don't need the CIP for the membrane chromatography, and it's feasible to use pump instead of a skid. For the TFF optimization, we were able to use cross flow that is more provide automation of the optimization. For the small scale purification, we use a high super miniature purification that help us to quickly screen the resin conditions, and batch purifications also help us to determine the PI of the VLPs. Also, with the one technology we use for it's called the AFA license. It's a small scale, very miniature scale of the license that help us to do the high size throughput screening for the license conditions. And since the viral-like particle is a very complicated molecule, so we have to rely some of the analytical tools using mouse and SDCs. Then we were able to obtain the knowledge of the size and determine what is our targeted antigens and impurities. So this is the high throughput small scale lysis technology we mentioned. Typically in the past, we either use a dynamill or microfluidizer to do the lysis. And if we want to do a buffer screening, we request our upstream team to generate lots of cell paste. And it requires a large amount of buffer preparation. And usually the sample size is also pretty big. So it's very time consuming and material consuming. So the team has putting together a beater, which it minimizes the sample handling to 2 hours, and the sample size from several liters to several milliliters. And there's one innovative technology called AFA. It's a focused ultrasonicator, and they can process samples 2 minutes per samples, and the sample size is 1 to 2 mils. So a good case study that we were able to screen our lysis buffer condition with 3 buffer components with the ratio and time, and just within 1 day. And the efficiency is largely improved. And when we use this same lysis conditions, when we scale up, it was a very successful scale up result that we'll be seeing. Also, our VLP, because it's big, so when we do the IEF gels, it cannot go inside the gels. So it's very hard to us to determine the PI of that VLPs. And if we don't know the PI, as most of the purification scientists know, it was pretty hard to determine the elution binding condition for most of the chromatography steps. So instead of that, we use a pretty simple setup using the 96 well plates, and we actually finishing all the pH and conductivity binding screening observation within 2 hours, and find the PI of that VLPs. And usually, if we're gonna run this for use a regular run on the extra, for example, one column, one run, it will be a lot more hours that we'll be seeing, at least 12 times more hours than we will be implementing instead of using this technology. So that was pretty exciting when we tried to implement this technology from all our resin screening and the condition screening. And then just need to confirm with one run on the large scale. Also, like I said, if we want to making the purification process more continuous, one of the technology we try to implement is using depth filtration for the yeast cell from the inclusive cells after disruption. So the vendor was helping us to do lots of depth filtration screening. As you can see the data on the right, we did a lot of screening, and most of them cannot handle this high density and lots of impurity feed stream very well. There's a few combinations that work, and we were able to achieve pretty good high throughput on this process, and then enable our scalable process and continuous instead of just using the ultracentrifugation technology to be able to clarify those cells. So it is a significant turbidity reduction, and it is a very high throughput technology. So when we screening our depth filter for the downstream, so we use canine change chromatography membrane. By screening all this canine change resins versus membrane, we decide to go with the membrane. First, it's a flow-through mode, and second, it's a single use, and we can attach them to the depth filter. So on the right side of the image, you can see this is a 4-train. Only request 2 hours to finish basically 3 steps. First step is filtration, second is the filter, 0.2 micron filter, and the third one is actually our chromatography step. So the time and the money we were able to save, we can see there's a significant saving on the cost as well. If we are doing this with the 1 cycle versus 100 cycles, we're still seeing a pretty significant saving. And with this process, within 2 hours, we can see the load starting with lots of cell debris, very turbid. And within 2 hours, we can see the pool is pretty clear, and the very low turbidity is, the purity is also pretty good. So that's enable our next purification step has less challenges, and the whole VLP process is actually pretty efficient. So this is a summary that gives some outcomes. So why we need to implement all this innovative technology. To some degree, it becomes disruptive. This is a perfect example to show that for our innovation enablers, we use single use the QBD to control the process characterization, and then we use lots of high throughput automation as well in different various steps. So our first E. coli VLP process in the current commercial manufacturing takes 2 weeks. And after we implement all this innovative technology, it does become very disruptive because it becomes a 1-day process. And the significant time and effort was reduced by doing a very robust process to deliver a high-quality product. So we're really happy with this result, and we do believe those innovation technologies can involve our manufacturing process by implementing that way. So the next case study, we're going to talk about the high capacity capture resin for the MABs. So as a process scientist, we're always dealing with which resin we're going to pick, we're going to screen, and we always think about how efficient the process is. So this is some question we always facing, and we basically become our daily task. So I want to give some examples. How do we do the resin screening and to think about the cost and the efficiency early definitely help with the late stage manufacturing process. So for most monoclonal antibodies, the challenge for the purification come with 2 things. Usually when process scientists receive some early stage transit cell harvest, the titer is actually pretty low because it's a very early stage. When also you receive a low volume range. So with the manufacturing process involving, you basically see a high titer and the volume is getting bigger. So your feed you have to facing a feed that involves different titers and different volumes. And the desire is to minimize the total cycle, shorten the time, also improve the quality. So we need to check first, do we get high dimer binding? Do we have a shorter resin time and a faster flow? Do we have better selectivity? Is the column easy to pack? The quality is good or the cost is good? So that's our check box that we need to cover when we do the resin screening. And this figure is showing we resin a bunch of protein resins, and we compare with the capacity, the pool volume, and the pool concentration. The pool volume tells us the story if our next process will be a much shorter time. So we look out at these factors, and we also double check the quality of the pools, which shows the comparability. And based on that, we basically looking into the production, which is grams of MAB per liter of resin per day. And we select the resin that offers the highest productivity and lowest cost, which is a very significant processing efficiency improvement and cost improvement. So my last section will cover how do we involve in the manufacturing process by risk mitigation. Basically, when we do the manufacturing process improvement, this is the workflow we typically will see. So first we write a study plan that covers small-scale process improvement, and then we will do a small-scale optimization study, and then we will do consistency run that to confirm it is robust enough and the quality is ensured. Then we scale up at the scale development run and engineering, write the development report and the technical transfer to our commercial teams. Technical transfer validation run will be need to write the protocol, manufacturing batch record. So this is basically a very typical workflow that we'll be seeing. So all these changes that we try to make, we will start from ground zero, which is starting from small scale process improvement. Then we will involve in the process the same way that as if we introduce a new process. This is one example for how do we optimize the wash conditions. And basically we use the DOE. We know where is our target hotspot and what is our risk. And we try to minimize the risk by picking the optimized conditions to maximize the quality and the quantity. So since we do lots of process improvements for our current commercial manufacturing, so one thing we want to make sure is our new process consistent or does it bring additional risk? One thing we can check is to look at the data analyzed from all the past runs. So this commercial runs has at least 10 years of commercial manufacturing batch record. So we look at all the data on the process and yield time. Also, we compare with the quality and the purity and activity of those lots. Then we will know our limit. At least the new process doesn't introduce any additional risk to the process. And we can see the improvement at least from the process efficiency side, if not on the quality improvement. Usually we also will see a lot of quality improvement for the process as well. So this is a summary of what we can do, what helps when it comes to innovation. We can adopt the solutions that potentially can improve the quality, lower the development cost, and reduce time to the market. We need to be proactive, educated to all stakeholders, because it's not only process scientists and the manufacturing folks have made the decision of implementing innovation technologies. We also need to involve the quality, compliance, regulatory team to buy in the ideas and see the benefits when we implement the new technology. We need to be collaborative, involving the vendors and academic and biopharmaceuticals, because it's not only one person can make the changes, the whole industry is making changes. So by actively talking to them, going to different workshops and meeting with people, talking about new technology, give us ideas, and work together, the industry is improving to the next level. We also need to minimize the risk, because if there's a lot of foreseeable risk, it has a high potential that will be the process of the new technology will be killed from the beginning. So we need to leverage our knowledge from the existing process. So we are not totally working away from the process. So we just try to improve that. And finally, I want to thank you for all the teams that help to implement all those new technology, including my past colleague from Grifols and Thomas, all the vendors, and from GE, Paul, MilliporeSigma, and some official covered. So thank you for the talk. I want to finish my talk. Thank you for listening to my talk. I want to finish the talk with a quote that, what would life be if we had no courage to attempt anything? And I will leave the question to the end of the workshop after John finishes the talk. So, thank you, everyone.
Thank you for that, Yuyi. Once again, I would like to remind our viewers that there will be a live Q&A session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second speaker. John Cyganowski is the Director of Manufacturing Sciences and Technologies Downstream Americas for MilliporeSigma, a business of Merck KGaA, Darmstadt, Germany. This is a team of engineering subject matter experts that assist in scale-up, validation, implementation of MilliporeSigma technologies, executing technical work for mid to late stage projects. John has specialized in tangential flow filtration, aseptic processing, viral clearance, and large-scale bioprocess manufacturing over his 25 years with EMD Millipore. He has served as an instructor for the Tangential Flow Filtration section of the annual ASME Downstream Bioprocessing and Economics course, and is a member of the Parental Drug Association. He is a member of the American Chemical Society Biot Division. He has served as the co-chair of Non-Chromatographic Separation section and as co-chair of Downstream Processes, Technology Transfer, Scale Up and Scale Down for the 2015 and 2016 ACS Annual Conventions. John holds a Bachelor of Science degree in Chemical Engineering from the Northeastern University. I will now be handing over to our second presenter. Welcome, John.
Hi, can everyone hear me okay? I just wanted to say thank you to Yuyi. That was a really nice presentation, and I'm looking forward to the Q&A. So today I'd like to talk to you about bioprocess intensification using single-pass tangential flow filtration. This has broad applicability both in batch processing and in semi-continuous or continuous manufacturing. So today, on the left-hand side of the screen, we can see the agenda, talk a little bit about background, and then some other areas where we think that single-pass tangential flow filtration, or SPTFF, could be utilized to make those steps more efficient. So on the right bottom-hand side of the screen, we see a typical batch tangential flow filtration system. There's a feed tank, feed pump, a holder to interface the membranes with the piping. There is a retentate stream that is recycled back to the feed tank and a permeate stream. So typically these systems are used in product concentration. We want the protein of interest to be retained by the membrane and to stay in the feed tank as we withdraw buffer as permeate. In batch mode, it requires multiple passes through the assembly, and it requires more equipment. At the top right-hand side, we can see a diagram of single pass, or SPTFF. The first thing you notice is that there's a lot less equipment. We think most of this equipment already exists in a plant. You really only need the holder and piping to be able to direct the streams. And this is where concentration occurs with only one pass through the filter assembly. So it's more or less it is a continuous operation, and you will need a holder. But because you don't need a lot of the ancillary equipment, this is easily retrofittable to existing processes. And you don't have to wait until you implement a continuous process. So, move on. So, let's talk a little bit about how SPTFF works. Essentially, it works by increasing the residence time. So there's a couple of ways you can do this. You could lengthen the feed channel. So you could have 3 sections of one module. So this gives you 3 times the residence time at the same feed flux. Or you can slow down the feed flow rate by adding membranes in parallel at the same overall feed flux. But the feed flux is reduced to 1/3 as it goes through the module. So again, you get 3x the residence time with the same path length. On the right side, you can see some experiments with high and low concentration SPTFF, and we're looking at conversion, which is the amount of permeate that goes through the filter divided by the flow rate that enters on the feed. And you can see these curves, they basically correspond to 3 different sections of membrane. So you can see that as you increase the residence time, you're also increasing the final the conversion of feed to filtrate. We can also see that, with 3 sections of membrane, you pretty much get what you're going to get. You don't really often see very many benefits of adding a 4th section. So typically we're talking about 3 sections in series. So here's an example of how single pass filtration might look in a laboratory or in small-scale clinical or large-scale manufacturing. So on the left-hand side, we can see 3.11 square meter cassettes. They are plumbed in series, and the way we do that is with these diverter plates. So you can get these 3 modules in one section. You could also do it with 3 different holders, but doing it with the diverter plates keeps everything pretty compact. The middle picture shows a pre-sterile tangential flow filtration capsule, and we've plumbed those in series just with the lattice fittings, the fractional fittings between the modules. These, because they come pre-sterile, offer the possibility of closing the UF process, which is good news for the antibody drug manufacturers and the vaccine manufacturers. And then for very large-scale implementation, the same hardware that is used now can just be set up a little differently, and you can get series filtration, within one level, and you can also plumb it so that one level then feeds the next level. So nothing really new here, except maybe the capsule device. Okay, so now that we know a little bit about single pass TFF or SPTFF, let's talk about some locations in the process where we think it would be useful. So here's a slide by Kaltenbrunner and Yuhan from the Accelerated Biopharmaceutical Development at San Diego in March of 2009. And here they looked at different aspects of batch processing, tank sizes, buffer volumes, membrane area, and then they show on the graph, on the chart on the right-hand side, up above, the x-axis shows increasing titers in the bioreactor, and then at the extreme left we can see a probability to fail color code. So basically, what we see is titers go up. At some point, either the batch won't fit in one of the vessels, or you won't have enough membrane area to accomplish your task in the amount of time that you want. So in general, we're talking about anywhere where there's membrane, so clarification, viral filtration, UFTF, and then the pool volumes. Typically the tanks are less than 50% of the working volume of the bioreactor. And then, buffer volumes. So this sort of presents the case that, okay, as your titers go up, we need to find ways to deal with this. Here's a slide from the folks at Biogen, Matt Westerby. Presented this at ACS National Meeting. So here we have an example with a 2,000-liter harvested cell culture fluid. Cell culture fluid. Here it's harvested with centrifuge, then it goes through depth filtration, and you get about 2,000 liters, 2,200 liters coming off the depth filtration. So here he's looking at using a single pass TSF system, really to decouple the harvest step from the process. So either to pool your harvest pool until you get enough material to run on the large process, or trying things like peg precipitation, so where you could recover the protein as a crystal, potentially store it or wash it, and then resolubilize as a purification step. And on this particular example, he's estimated that for a 30x concentration at half a liter per minute per square meter, he would need 10 liters per minute on 20 square meters of membrane. So not very large, and potentially retrofittable in a plant. So this could go in, and you could leverage the benefits of single pass in an existing facility, not just a new process. So, there's probably a lot of questions here. Okay, what does this pre-concentration do for my product quality? So here there's a chart here which shows SEC results. So you can see the reference SEC result for the protein, and then looking at post single pass, you see really that not too much happens to the really protein quality is unaffected by the concentration. step. And then the PEG purification pool, so doesn't change much on the high molecular weight, but it's mostly monomer, and they don't really see any low molecular weight proteins. So here's an example of SPTFF implemented on, say, a harvest step. And again, what we're seeing is that, looking at conversion is a function of feed flow rate. As you decrease your feed flow rate, you're increasing your residence time. So you're getting higher and higher conversions. Now, one of the things we'd like to bring up is that, some of these high conversions can show flux decline. And usually this is because of impurities, concentration in the harvested cell culture fluid and precipitation on the membranes. This can be a real problem if, this problem emerges usually with very high cell densities and poor viability, simply because there's a lot more cells there and a lot more debris. But if you have cells which are, the viability is high, then this shouldn't be too much of a problem. So some of the things you can do to manage this would be to limit the conversion. You could use a depth filter on the harvested cell culture fluid to polish the feed, and you can add extra area to allow for membrane valve. Okay, so we've covered tank debottlenecking, and now we'll talk a little bit about the capture preconditioning, how it can enhance productivity and capacity. So here we see a breakthrough on this MAB05. We used Ishmuno A, protein A resin. This breakthrough curve is for, higher concentration and lower concentration protein. And as we can see, that increasing the concentration of the feed doesn't significantly affect the resin capacity in the breakthrough curve. However, increased residence time improves capacity and sharpens the breakthrough curve. So taking that data, this MAB05 data, we did some computer simulations with BioSC software platform, and we modeled a continuous process where I think there was it was looking at pre-concentrating and potentially the number of columns that you might need. So in this simulation, essentially what we're suggesting is you could stay at the same mass flow rate, right? But as you pre-concentrate your feed, your flow rate is slowing down and thereby increasing your residence time. So the column is just as productive, but you have increased residence time. And then here's some times and assumed times for some other chromatography steps. So here we've got productivity and residence time, or number of columns, plotted against feed concentration. So in this particular column, we can see that as you increase the feed concentration, you start to get higher productivities, and not at not at excessive residence times. And we can also see that as we increase the feed concentration, this has an effect on the number of columns that you might want to use for this continuous process. And here we see another column, the different bed height, and we see the same sort of effect. And we think that's pretty important because a lot of these continuous processes, people are looking at 3 columns. So we think it's possible to do it with 2 columns if you pre-concentrate the feed. So it'll increase the productivity, reduce the number of columns. The benefit's more pronounced when the columns are small. But by reducing the load flow rate, it reduces pumping requirements and could also potentially enable small systems to process large batches. So more efficiency. Okay, so now we're going to spend a little time talking about the polishing and preconditioning step, how you could enhance productivity and capacity and viral safety. So here again is another slide from the folks at Biogen. This was also at ACS National Meeting in 2012 where they presented this. So here we have, I think, the protein A pool. Having a hard time seeing that. Here I think we have the protein A pool. It's about 2,700 liters. Now because it's coming off the protein A, and the TMAE resin is not self-tolerant, you need to take a dilution. And so you increase the size of the pool that has to go onto the column. And then with recover and everything, you're up over 9,000 liters, which is important if you have constraints on that vessel. I think the problem statement was here that they didn't want to see the volume go over like 8,000 liters. So here's the same step, but this time with an SPTF step inserted between the column and the protein A virus inactivation pool. And here you pre-concentrate this by about 4x, same size batch. So now you're down to 715 liters. Do a 2x dilution. And now you only have a pool that's like 2,100 liters, and you pick up a little again in the recovery of the column. So this is of real interest, and you can see some specifications here, or some process parameters here. So large-scale manufacturing tank volume. So they have a limitation. So you can see with pre-concentration, you can easily fit these pools within your tanks, and you don't have to change out a tank and revalidate. It's just the same equipment that you've always used. We got really high recovery with a single flush. We don't see any significant impact in product quality, so the pre-concentration didn't really affect the high molecular weight species, and the purity of the protein was also similar. So now I've convinced you that the pre-concentration doesn't affect protein quality, but now you're probably asking, okay, well, what does it do to my host cell protein? So here, this is another thing that we could do to boost the performance of a Q column to clear HCP. So in this case study, we used SPTFF and Ishmuno-Q in the flow-through mode. We conducted these separately for individual analysis. Essentially, this was an experiment where we looked at different feed rates, and again, you can see as you go to lower and lower, or longer and longer residence time, getting a more concentrated feed in the retentate. Now, the HCP does not permeate the 30 kilodalton SPTF membrane. It co-concentrates with the MAB, and this will provide improved AEX clearance. So here is a sort of a, you can think of it as an isotherm type of curve. And here we're looking at different concentrations of HCP we achieved with SPTFF. And you can see that the more concentrated the host cell protein, the higher the dynamic binding, or the higher the binding capacity is for the host cell protein. So, the pre-concentration is actually exploiting the shape of the isotherm and providing you with enhanced capacity. Let me build up this slide. So, so here's an experiment where we looked at host cell protein in the pool, and we looked at MAB loading on the resin, on the Q resin. So here's a curve at 11 grams per liter, and you can see, you're probably getting, if your limit is 10 ppm of host cell protein, you're probably getting that with a loading of somewhere like 125 mg per mil of resin. So then we ran a more concentrated step on the column, a more concentrated feed on the column. So here we've tripled the amount, and by the time you get to 10 ppm, you're probably like maybe 150, 175 mg per mil of resin. And we continued this for a couple of other concentrations. And you can see that the more you concentrate, the better your host cell protein clearance gets, up to about 600 mg per mil on the resin. So it's making your column much more productive. Now here's an experiment that we ran. The red curve is at 99 mg per mil, and we see the same shape of the curve, but it's certainly offset backwards. And we think that you're getting into an area where you've got a lot of host cell protein and product protein interaction. So there is an upper limit to where you want to concentrate. So we think that the SPTF, we've shown that we can boost the loading on Shimano Q by a factor of 4. We think that we can increase the productivity by a factor of 5x. So it could reduce process load times and make the step much more efficient. Here's a look at some small-scale equipment setups. So we have a pump where you can take all your different solutions, your buffers through the single pass if you want, but your protein solution can be pre-concentrated. There's a static mixer, and then it kind of goes through the column. So very, there's pressure gauges, and there's like a bubble trap. Very simple setup that's for testing. Now, you can even use this in a continuous process where you wanted to cycle back and forth between columns. So you can do an inline dilution of high conductivity feed, and you can use this multi-column rotation. So you would be processing over 24 hours. So we believe it's possible for continuous processing applications. So I've probably convinced you of the protein quality is not affected and that we can clear more HCP. but you're thinking, well, what about viral clearance? What happens to my viral clearance? So here is a experiment. I think we did MAB1, and we spiked it with MVM and XMULV. We used the Ashmuno Q column. Again, that's the resin that we have. And now we're going to look at LRV of the pool based on loading on the resin. So what we're seeing here is that, so here's the 11 gram, the blue dots are the blue squares are the 11 gram per liter protein, and then the green ones are 79 grams per liter. You can see viral clearance is, not really affected by the concentration. You get the same high clearances that you would expect in a more dilute process. And here are the XMULV results. So maybe, I don't know, maybe a little bit better clearance, but very, very similar. Okay, so I'll have one more slide for viral filtration, and then we'll wrap up, and we'll get into the Q&A session. So here's a figure from one of Glenn Bolton's papers. Glenn is with Amgen. He looked at this a few years ago, so he's looking at different viral clearance filters and their capacity as a function of protein concentration. So we can see is there's a couple of these filters. So the CPV and the Planova 20N, their capacity curves are definitely a function of protein concentration. Now, one of our old viral clearance filters, the Virusol NFP, also had a very strong relationship to, its loading had a very strong relationship to protein concentration. You're really limited to about 10 grams per liter. But the new technology filter, the Virusol Pro, we can see that capacity goes up. Now, this is going to be specific to what filter you use and what molecule you're using and how pure it is. But we think, we could potentially use SPTFF to reduce the total cost of the viral filtration. Okay, so I'm going to do a quick summary, thank my colleagues, and then end the presentation. So essentially, single pass tangential flow filtration intensification. You can do pre-concentration by UF using higher residence times. They're simple systems, simple operations. You get high recovery, low minimum working volume. Reduce protein pumping, so potentially that would help your your protein would be unaffected by the pump passes and potential denaturation. It's run as an inline operation and has a very small footprint. So our SPTFF technology, Millipore's technology, uses existing hardware and cassettes, which means that you might already buy these cassettes. It's just a different way of plumbing them together. It's useful for tank debottlenecking, intensified capture, intensified polishing, and intensified viral clearance. We can implement this with either batch or continuous processing. So the overall results, this is a quote from Biogen, demonstrate the feasibility of SPTFF as a platform purification technology. So finally, I want to recognize a few people. Andrew Brinkman, I think his name is, and Kaltenbrunner, and from Amgen, and Teska, who wrote an article on this. And so then I want to also thank my colleagues, Tom Ellick, Dave Bohonic, and Herb Lutz. And here we are.
Thank you for that presentation, John. Now we will begin the question and answer segment of the webinar, But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. And now for our first question. What is considered disruptive technology and why do we need them? Yuyi, would you like to take this question first?
Okay, thank you, Stephen. Thank you, John, for presenting this awesome technology, which you use down the road. I think this is sort of, if we got implemented into the commercial manufacturing, it will become disruptive. So I think there's no recognized definition for the disruptive technology in bioprocess. So everybody has different view on this innovation technology is disruptive or not. My understanding is, like I said, it will change throughout the life cycle of the product, because early on you make small changes, even though it sounds like it's a brand new technology, but it doesn't got implemented, and it doesn't have a very significant impact. So it does vary with the stage of the product. When it's getting close to the later commercial manufacturing, even a small changes will make a big difference. So most of them, I think, it's from the incremental improvement. So the disruptive impact will come after the fact that you combine it within all the factors. Some of the examples I would think, the membrane technology that we mentioned, the single-use technology, and continuous downstream, and the technology that John just mentioned with the single Pass TSF that's making our continuous downstream combined with the upstream, the precision bioreactor. That will be a very disruptive technology for the whole industry.
Thank you for that. Our next question is, how does a PD team get started with single-pass tangential flow filtration? John, would you like to answer this?
Sure. So as we saw in the presentation, the equipment is rather simple. MilliporeSigma, our technology is based on the standard pellet contact sets with the same catalog numbers that you already use. If you have data generated for TSF, batch TSF operations, this data can be utilized to optimize the operation of a single pass. The user and validation guide information remains current. It's unaffected by the operating mode. The supply chain for ordering these may already be in place, especially if you already use this technology. It's only the operating method that really changes. So you may wish to purchase diverter plates, or you can use multiple holders to create sections in series. And you need some way of driving the fluid, so either peristaltic pump or use a pressure vessel to drive fluid.
Thank you. Our next question on the back of that is, we've already tried SPTFF and it didn't scale well. Why do you think your devices would be any more scalable?
Yeah, so that's a good question. So our approach to single pass is to use the standard devices that we already have. They've been in the market for a long time, and we've scaled these from like 500 mils to thousands of liters for batch processing over years. So we have a good track record there. Because we use these same devices in single pass SPTFF operations, the built-in scalability is really the same. And we have demonstrated SPTFF scale up from like 1 liter to 500 liter with predictable and reliable results. So you won't get disappointed when it comes time to scale with this approach.
Our next question is, can you provide advice for the scientists who want to implement innovative technology into their process? Yuyi, would you like to answer this question first? Or John, if you have something to add, would you like to answer this? That's can you provide advice for the scientists who want to implement innovative technology into their process?
Sure. So obviously, everything begins with a scale-down process. You want to try to study the unit operation at the small scale and prove out the concept. And then certainly a pilot scale operation to demonstrate that the technology can be scaled. I think it's say not just a PD decision. I think it's a kind of a group decision where you definitely want to work with your quality assurance folks and you know, determine sort of the best way to implement the new technology.
Dr. Shen, would you like to answer this question as well? So that's, can you provide advice for the scientists who want to implement innovative technology into their process?
Yeah, I kind of agree with John that it involves lots of stakeholders, so you do need to work with them collaboratively that build trust and a good partnership with them to establish some models, including all the critical aspects, the concerns, all the risks list ahead of time, discussing with the PD scientists. Also, we will be encouraging thinking about the more high throughput technology to save time and cost. There will be some gaps to recognize the volume to actually make it work. So my piece of advice for the PD scientists and manufacturing folks will be keep your faith, good faith in believing this technology, request your persistence when it comes to it, because there's always some roadblock, like I mentioned, there's many of them. So have a good trust and faith into the innovation, and the rewarding will be very significant. It will eventually benefit the industry, our patients in the end.
Well, thank you for your answers. And with that, we will conclude the question and answer session. Thank you, everyone, for the questions that were sent in. Before I finish the webinar, I would like to ask our presenters, Yuyi and John, if they have any closing remarks. Yuyi would like to go first.
Yeah, I first would like to appreciate this opportunity to share with my presentation with all the scientists and manufacturing folks here. And like I said, I would encourage when we implement the innovative technology, have a faith and work very closely with all the stakeholders to make sure it happens, and believe the results will be very rewarding. Thank you.
And John, do you have any closing statements or final remarks?
Sure. I would like to thank the audience for participating and although my portion of the talk was the vendor portion, but hopefully I showed enough data here to make you feel like you know, this is real and it's not a commercial. The other thing I would like to say, it's a very exciting time here in bioprocessing. There's a lot of innovation, innovative thinking going on, and innovative technologies coming into the market. And I mean, this is a chance to improve things. So I would embrace the new technology and not be afraid of it. I think I have a question from the audience. So it says, in single pass TSF, reducing the flow rate can change the delta P value. So what can be ideal delta pressure? That is true. You can change the delta P value. But as we know from just standard TFF development, if you look at flux versus TMP, you know, you get the curve, the hockey stick shaped curve, with the optimum TMP somewhere around the knee of the curve. It's the same thing with SPTFF. So although you reduce the flow rate and change your delta P value, you can tighten down on the retentate valve. So you can maintain your optimum TMP. The delta P will be smaller, but your TMP will be right where you want it to be.
Well, thank you for that, and thank you for answering that that last question there. I would like to take this opportunity to, whilst we have time, to thank our presenters, Yuyi Shen and John Cyganowski, for sharing their knowledge with us. And I would also like to remind our viewers that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand webinars. And lastly, if you are watching this on demand or you have another question, please send those over to stephen.edwards@biopharma-asia.com, and we'll get those answers to you. Thank you, everyone, and have a great day.